Hi all, Dr. Clark again. General biology lecture today. We're going to talk about prokaryotic and eukaryotic cells. And we're going to talk about something called endosymbiosis, or the link between prokaryotic cells and eukaryotic cells. Okay, so I'm going to talk about the theory of endosymbiosis, which suggests that, with lots of evidence, clearly, um, that prokaryotic cells gave rise to eukaryotic cells through the form of symbiosis or endosymbiosis. Okay? Kind of what's being depicted here, okay, that phagocytosis would occur and some small prokaryote would be engulfed, and then instead of being dissolved, it is utilized. Okay? So like a photosynthetic prokaryote would be then utilized inside of the cell instead of breaking it up and dissolving it. Instead, it's utilized, and the energy can be then transferred into the cell. And so we'll come back to that in a little bit. Okay? But first, we're going to talk a little bit about the two types of cells. Okay? Prokaryotic cells. Prokaryotes meaning no true nut or no true core, refers to the lack of nucleus. Okay, so these cell types do not have a nucleus. Now, just because they do not have a nucleus doesn't mean they don't have DNA. They do have DNA. They have the capability of having RNA and DNA and pretty much all the features of cells that have a nucleus. It's just they have no membrane around their DNA, no separate membrane around their DNA outside of the plasma membrane. And I'll show you an example of this in a little bit. Okay, so this includes the two kingdoms, archaea and bacteria, which, as we already discussed, are also in their own domains. So you have the archaea domain and the bacteria domain. Okay? And probably when all said and done and everything's been classified, these are probably, at least bacteria for sure, is probably the most species rich. So they have the most species in any kingdom that is out there. Okay? It's just we haven't got around to naming all the bacteria per se, and they evolve really fast, so I doubt we'll ever name all the bacteria, but nonetheless, very species rich, so lots of cells on the planet have this kind of characteristic. Eukaryotic cells are the cells that most people are familiar with, okay, because these are the types of cells that we have, and that means they have a true nucleus, okay, and also, along with that true nucleus, which is membrane-bound DNA, you're going to have other membrane-bound internal compartments, which might have multiple different functions. Maybe it's the site of ATP synthesis. Maybe it's the site of photosynthesis. Maybe it's the site of digestive enzymes. There's lots of different functions that could be around these internal compartments. Okay? And then when we're looking at this, we're talking about the other four kingdoms. Okay? So we're talking about Protista, fungi, Animalia, and Plantae have eukaryotic cells. Okay? So first, let's look at the prokaryotes. Okay? Simple cellular organisms, okay, and they are the simplest. Okay? And simplest meaning that the amount of internal compartments and things like that that they have, and the functionality is simple or simplistic. It doesn't mean that they don't have complex relationships, because they do. Okay? And so if we look at them, there are a lot of similarities between prokaryotes and eukaryotes, and we'll talk about the similarities and then what are the differences. Okay, so first, they have a plasma membrane. Right? And that plasma membrane is just like the plasma membrane that we talked about before. It's going to be a phospholipid bilayer. Okay, you're going to have embedded proteins. You might have some cholesterol. You're going to have these proteins that are transmembrane proteins. They run all the way through the membrane, which allows for things to flow in and out. You might have cell surface proteins, which allow for the cell to be recognized by foreign bodies, either as being self-made or something that they can communicate with, these kind of things. So other cells can interact with them, especially in the colonial cells, where you might get biofilms of bacteria being made. They need to interact with each other, and so they might have these outside proteins, transmembrane protein, or cell surface proteins that allow for that connection. Okay. They have cell walls now. The construction of the cell wall, what it looks like, what the cell wall is made out of, okay, is different than the cell wall of, say, plants. Okay, but nonetheless, it's an extra layer of protection around the plasma membrane. And some bacteria, some prokaryotes, will have an additional layer of protection, which we call the capsule. Okay? And this will play an important role when we start talking about antibiotic resistance, when we start talking about whether something is gram-positive or gram-negative. Okay? These are tests to see if that prokaryote has that capsule. There's other tests, okay, like capsule tests and acid-fast tests and other tests that can indicate whether or not a bacteria might have a capsule or not. And it's just an extra layer of protection that some bacteria will have. Okay. So other than that, the cells are pretty simple in organization because there's no internal compartments that are membrane bound. They do have occasionally a little bit of protein work and protein fibers that hold the structures together, okay? But it's not a support framework like we're going to see when we look at eukaryotes. Okay? And because all living organisms have, some information carrying molecule, either DNA or RNA, there has to be a way in which that molecule can be used to form proteins, because that's the purpose of DNA, is for protein synthesis. Okay, so all prokaryotes will have ribosomes, which means they have their own ribosomal RNA, and this has allowed researchers to make those connections between eukaryotes and prokaryotes, or eukaryotes and archaea and bacteria, to kind of get an idea of, well, when did archaea and bacteria arise on the planet? How many mutations have been formed since then? What's their link to eukaryotes? And this can all come because all living organisms have ribosomes, which means all living organisms have ribosomal RNA. And we'll come back to this when we talk about the function of ribosomal RNA, when we talk about DNA synthesis. Okay? Prokaryotes will also have a nucleoid region. Okay? So they do not have a nucleus, but they have a region at which you would expect to find the DNA. Okay? And so the DNA is in chromosome form, and it's in a circular chromosome. So instead of linear chromosomes like you see in eukaryotes, it's a circular chromosome that the ends are connected. Okay, and we'll talk about, when we talk about how DNA is copied, and we'll talk about the benefits of having that circular chromosome versus linear chromosomes. But nonetheless, the nucleoid region is where you would expect to find the DNA. It's also the region where ribosomes are going to be constructed and synthesized. Okay, and but again, there's no membrane there except for the plasma membrane that goes around the entire cytoplasm. Okay, there are other structures that prokaryotes have that can be unique to prokaryotes, but lots of eukaryotes have the same capabilities or similar functions and similar features. Like flagellum, okay, eukaryotes also have flagella, which are long protein fibers that allow for movement. Okay, they can be associated with moving the organism through liquid solutions, but also can be associated with feeding. So they can beat their flagellum and cause kind of a current, and that can go into kind of a feeding apparatus or feeding groove that allows for them to consume food with that flagellum. I'll show you a picture of flagellum, but if you know kind of what a sperm looks like with a long tail, that essentially is the flagellum of the sperm. They also have kind of unique structures called pili or pilus, which allow for some prokaryotes to exchange small chunks of DNA called plasmids. And plasmids are unique circular chunks of DNA that tend to mutate fairly quickly, and they also tend to be very adaptive. So when bacteria are subjected to like high temperatures or high pHs or high salinity or antibiotics or these different things, the plasmid region will often mutate fairly fast, and then that plasmid region can be passed from one bacteria to another bacteria, and it's easy for them to pass it between within species. Okay, but even more than that, a lot of times those plasmids can be passed within genera, and sometimes even between genera. And so you can get antibiotic resistance in case of where a plasmid is involved. You can get that built up in not just the species you're after, but all species that are related to it, and even across some genera. Okay, we'll talk more about this when we progress with bacteria. Okay, but here you can see a typical prokaryote. Nucleoid region, okay, where you find the DNA. The DNA is in a single chromosome. Okay, flagellum used to propel this organism through liquid solution. Okay, it has 3 layers: a capsule, cell wall, cell membrane. Cytoplasm, cytosol. Okay, same thing. There it's showing the pili or the piluses. Okay, where DNA can be exchanged. It's not showing plasmids here, but you would have these little circular chunks of DNA that occur throughout kind of the cytoplasm. Depending on the type of bacteria, depends on how many there might be, and the capabilities. Okay, and then there you can see the ribosomes, the sites where proteins are going to be synthesized. So eukaryotes, okay, a little bit more large in size, but again, remember, based on the surface to volume ratio, eukaryotic cells are not very big still. They are a little bit larger than prokaryotic cells, maybe sometimes double or triple in size, but still it's small. Okay, and they tend to be much more complex than prokaryotic cells, and so we'll look at this complexity as we progress. Okay, but again, just like prokaryotic cells, they're going to have a plasma membrane, okay, made of phospholipids, proteins, these kind of things that encase the cytoplasm or the cytosol. Okay, but inside that cytoplasm, they're also going to have membrane-bound organelles or membrane-bound compartments that we call organelles. And each one of these might have a particular function or a particular job inside the cell. Maybe it's to make lipids. Maybe it's to digest food. Maybe it's to, process carbohydrates. Maybe it's to make carbohydrates. Package things. There's lots of different situations, lots of different organelles out there with different jobs or purposes. Okay. Again, in the cytoplasm, more so than prokaryotic cells, eukaryotic cells are going to have protein fibers which make a cytoskeleton. And these protein fibers can be utilized as transport vessels, so like things can attach to the protein fiber and move from one organelle to the next. It can hold organelles in place, and it can keep kind of the structure of the cell from collapsing on itself. Okay? So when we look at organelles, though, some of them you can see under the microscope fairly easily, okay, like the nucleus. And I'll show you some pictures of this, and then if you're live lab, you're going to experience seeing nucleus. Okay, like I said before, eukaryote is the opposite of a prokaryote. Eu meaning true, karyote meaning nut, okay, or core. And so eukaryotes means true nut, prokaryotes means no or non-nut, and that's kind of where the name comes from. It's because of the nucleus. Right? And so the nucleus is just a membrane-bound compartment that houses DNA. Okay? Now, inside that cell, again, like I said before, you're going to have these internal membranes, which we call endomembrane, or the endomembrane system. Okay? And you can have all these compartments that have different purposes, provide very specific jobs to the cell, okay, that are membrane bound, called organelles. Okay? Now, just like all prokaryotes are not the same, all eukaryotes are not the same. Now, we didn't really go into the differences in prokaryotes, like the difference between archaea and bacteria, but we will later lectures. Okay? But the differences between some of the cells that in eukaryote are much easier to see with the naked eye. For example, cell walls occur in plants and fungi and some protista, okay, which means that they have a plasma membrane and then they have a cell wall that bounds or is on the outside of that plasma membrane. This often is made of cellulose, okay, a very complex carbohydrate. Now, some protista can have organelles called chloroplasts, which give them the ability to do photosynthesis, and all plants have chloroplasts in different amounts and different capabilities, and we'll talk about this as we progress. Okay. Plants contain a central vacuole, okay, and central vacuole is kind of like a storage vessel. It can store water, it can store nutrients, it can store ions, but it also can store digestive enzymes and is utilized for digestion often. Okay? And that's kind of unique to plants. Okay? And then animal cells can have centrioles and lysosomes, and these are kind of unique to animal cells. Centrioles will play a really important role when we start talking about mitosis and meiosis. These are the sites where microtubules originate from, and we'll come back and we'll talk about these a little bit more. Okay, so here you can just see the general structure of an animal cell. Plasma membrane, okay, and remember this is a fluid mosaic, so this is not rigid, okay, it has the ability to move, be pressed, take shape, flex quite a bit, but inside you have a lot of like scaffolding proteins, okay, that hold these organelles, often will hold these organelles in place, okay, so they're not getting banged around and moving around quite often, okay. There you can see the nucleus, okay, some other things like the endoplasmic reticulum, both rough and smooth, okay, mitochondria, Golgi apparatus, lysosomes, lots of things going on here, and we'll come back and look at some of these in much greater detail. Then you can check out the plant cell. Okay, plant cell. A lot of students don't see this, but plant cell, just like animal cell, has a plasma membrane. Okay, same phospholipid bilayer. Okay, it just happens to be on the inside of a very rigid cell wall. Okay, and other than that, there's a look at that central vacuole kind of storage vessel with enzymes. There's some chloroplasts that you don't see in animal cells, okay, and then general structures that are very, very similar between the two. Okay, so that really brings me to the aspect of how did eukaryotes evolve. Okay, so you have prokaryotes, which are very simplistic. Okay, basically DNA storage vesicles with the capability of synthesizing proteins, consuming energy, and whatnot, but they don't have individual compartments um, for their organelles. Okay, so even the bacteria that can do photosynthesis, they do not have chloroplasts. Okay, they have photosynthetic membranes. Okay, but no chloroplasts. Okay, so there's no membrane-bound photosynthetic pigments. Okay, so how did it come about? Through endosymbiosis is where most of the evidence suggests, uh, and we'll go into kind of how this works. But this, entity, this maybe prokaryote, is going to consume this prokaryote, okay, through a process that we call phagocytosis. But most of the time when this occurs, digestive enzymes would leak into this vesicle here, this capsule here, and destroy whatever's in here. And then this would be utilized as energy or treated as a foreign body, and the waste would be gotten rid of. Okay? But in some cases, okay, it doesn't happen. The digestive enzymes are not utilized, and instead this is encapsulated and it's maintained inside the organism. And so this then gets incorporated into the organism's daily life or whatnot. And if this is photosynthetic, then it would create carbohydrates and then pass carbohydrates across the membrane into this portion of the cell, and it could be utilized. Okay? And so the idea is that if these are beneficial bacteria or beneficial prokaryotes, that some of them would be engulfed and not destroyed, but utilized as powerhouses to the cell or photosynthetic pigment areas and whatnot. Okay? And there's quite a bit of evidence for this. Okay? And so when we look at some of the evidence, we see things like mitochondria and chloroplasts, okay, that have their own DNA. And their DNA is separate from nuclear DNA. Okay? And in fact, as a human, your mitochondria, you get that from your mother. Okay? And we know that mitochondria is what really gives you kind of the baseline of your metabolism. And that really comes from your mother. And your dad has no input on that because you get your mitochondria from your mother because you get the mitochondrial DNA from your mother, which is a circular chromosome, very similar to a prokaryotic chromosome. Okay? And just like plants, they would get both their mitochondria DNA from the mother plant and the chloroplast DNA from their mother plant. And so there's a little bit there. That's a little bit of evidence that suggests, hey, look, we have circular DNA. This is very similar to prokaryotes. But there's a lot more than that. Okay? Mitochondria are about the same size as modern bacteria. The cristae that we see inside the mitochondria, so these are like finger-like projections inside the mitochondria where electron transport system proteins are going to be kind of embedded, so ATP can be synthesized in those regions. They resemble the folded membranes in modern bacteria. Mitochondria have their own ribosomes, okay? And so because they have their own ribosomes, they can synthesize their own ribosomes. We can look at, structurally and genetically, are the ribosomal RNA of bacteria similar to mitochondria, and they are. Okay? Mitochondria, when they divide by binary fission, okay, just like a modern bacteria does. So instead of pinching off and forming a cleavage furrow, they just elongate until the membranes get really thin, and then they form a new membrane and disconnect. Okay? And a lot of this evidence, a lot of this material came from Lynn Margulis, okay, who I'm really admire Lynn's work and admire her scientific evidence for endosymbiosis, but not just that. She, being a woman who, was working on science when there wasn't very many women in science, and then also being ridiculed by a lot of individuals for her ideas. She wrote these 3 books and a lot more, but the best one, if you're looking at symbiosis, is A Symbiotic Planet. Excellent, excellent book. Um, but then after that, she really talked about how organisms can acquire genomes and acquiring them from other species. Talking about, kind of the 10% of our DNA is virus DNA. And then, what is life? And she is an excellent author. But more than that, she wrote the endosymbiosis theory. She wrote it up in a paper, and she was rejected by numerous scientific journals until someone picked it up, and it's thought as the piece of work that really drove the symbiotic theory. And now very few people would ever question that this is not the evidence for how prokaryotes gave rise to eukaryotes. And even more than that, how we got different genomes, and how we carry a lot of bacteria DNA with us, and protista DNA, and virus DNA, and all kinds of things. And her work was excellent. Great author. I suggest any of these. Doran Sagan is her son, and is an excellent writer also, and so is co-author on a lot of her books. But Lynn was an excellent biologist, and kind of drove the endosymbiosis theory, and gets all the credit for it. I mean, she did it. So, all right, with that, next time we're going to talk about specific organelles, and we're going to talk about nucleus, mitochondria, chloroplasts, lysosomes, central vacuole, and I'm going to show you some pictures of them, and just go into a lot more detail about what's their function, where they found, kind of what interacts with what, and then we'll progress from there.